This is TWIS. This Week in Science, episode number 873, recorded on Wednesday, April 27th, 2022. This show has teeth. Hey everyone, I'm Dr. Kiki, and tonight we will fill your head with death, regeneration, and fear. But first, thanks to our amazing Patreon sponsors for their generous support of TWIS. You can become a part of the Patreon community at patreon.com/thisweekinscience.
Disclaimer, disclaimer, disclaimer. Imagine a world without science. Not a world of yesterday, but a world of tomorrow. A world where all at once, while everyone was sleeping, the scientists and teachers of the world simply walked away, put down their pipettes, deleted their algorithms, left their students to languish. What then would the world of tomorrow look like? If in a generation there was no math, no biology, no chemistry, no logic, no critical thinking being taught. If research in every field simply stopped, what then would the world look like 5, 10 years? Global warming, no longer talked about. As sea levels rise and wildfires burn, the next pandemic just goes unchecked. Economies in ruin as hospitals staffed by volunteers of untrained people are overrun. The history of the planet in the hands of revisionists has always been authoritarian. The environment, according to industry spokespeople, has never been cleaner. The next generation, uneducated, raised on propaganda, unable to tell fact from fiction, with data, facts, truth all replaced by spun narratives, slick ad campaigns, and well-marketed conspiracy theories. In short, the world would devolve into what it nearly is today. And the only thing keeping us from this dystopian vision of a not-too-distant future? This Week in Science, coming up next.
I've got the kind of mind that can't get enough. I want to learn everything. I want to fill it all up with new discoveries that happen every day of the week. There's only one place to go to find the knowledge I seek. I want to know what's happening. What's happening? What's happening this week in science? What's happening? What's happening? What's happening this week in science?
Good science to you, Kiki and Blair.
And a good science to you too, Justin, Blair, and everyone out there. Welcome to another episode of This Week in Science. We're back again with all the science. Well, maybe not all the science, but a lot of the science. Well, a very well curated selection of the science from the past week that we all really enjoyed. And we are so glad that you're here to join with us to discuss, appreciate, and find curiosity. Whew, what did I bring for the show this week? I brought stories about all your base, snot bugs, fear extinction, and brainwaves. What did you bring, Justin?
I've got cellular regeneration, death rates amongst children, couple of looks back at the COVID strategies, attempts to curtail the virus spread, and apparently I've also got an indigenous fire suppression recommendation going on in Canada, and one more something too. What was the last? Oh, the one I haven't seen yet.
That one's gonna be great.
Megafires and microbes.
Megafires and microbes. Yeah, that sounds like a children's book of megafires and microbes.
Huh.
Blair, what is in the animal corner?
Scorpions, spiders,
Perfect.
tits, and bloodworms.
What have you done?
I don't know.
I am really looking forward to this show. The animal
boy.
corner is full of all the good things. Creepy crawlies galore. As we jump into the show, I want to remind you that if you are not yet subscribed, you can find us all places podcasts are found, pretty much. And you can find us on YouTube, Facebook, and Twitch, where we broadcast weekly on Wednesdays at 8 p.m. Pacific time. We are TWIS Science on Twitch, Twitter, and Instagram. Everywhere else, look for This Week in Science. Our website is twis.org. All right, time for the science. Do you remember that old song? All your base are, or I guess it was all your base are belong to us, and there was like a meme. It was an early internet
Is it a
meme about all
song?
your.
It's like it was from a video game.
Oh, it was a video game. Maybe that's what it was.
I thought you
Yeah.
were gonna sing all
All
about
your
that
base
bass.
are belong to us.
Also old, but not as old.
Well, all your bases, RNA and DNA bases, have been found in meteorites that have landed on this planet. Just published this week in Nature Communications. Researchers talked about their analysis of several space rocks and the nucleobases that they were looking for: adenine, guanine, cytosine, thymine, and uracil. And these bases are important. Does anyone get why they're important?
They
That's
make up the constituents of RNA and DNA.
Yeah.
Blair's like put together.
I don't know what
and
Blair's
putting-
doing.
They're the
Blair,
puzzle
that's not appropriate
pieces.
for the show.
So the G goes to C and the A goes to T and the
Yes.
yeah.
So
And
these
then there's the
nucleobases
U's that-
make up the chains
Yeah,
of DNA and RNA, and they cross-link with each other to make our chromosomes. And we really would not have life without DNA and RNA. But where did DNA and RNA come from? Did it just start here on Earth in the primordial soup? Well, this result specifically suggests that that's not the case. That all of the starting materials for the amino acids to create the proteins that work for life, all life on Earth, that they exist in space. And they were brought to Earth by meteorites.
Oh, okay. So I want to
That's what it
clarify.
suggests.
Life did not come from the meteors. DNA
That's
did not
not-
come from the meteors.
Yes. No.
Just this individual piece of DNA. came
All
from
of
meteors.
these individual
blocks.
parts of DNA,
Right.
all the little pieces, the adenine, thymine, uracil, guanine, cytosine, all of these have now been found on meteorites.
So it doesn't mean
So-
life came from outer space, but it does mean that potentially
The building blocks.
the building blocks came from outer
Yes.
space, and it would not have been possible to have life without those building blocks that came from outer
Wait.
space.
Yes.
No, does it mean that, or does it mean that those building blocks also exist in space
Or
as
that.
a natural occurring thing?
The building blocks could exist in space. They are these meteors. They fell to Earth, and they had been in space previously. They fell to Earth containing these bases, and the team has tried to figure out, did this stuff just end up on these samples once it
Oh, sure.
landed on the planet? But no, they think that the detected chemicals and the way they analyze them, that it is compelling to support the hypothesis of interplanetary origin for the building blocks of life.
Okay, so they're also sure
Origin.
it's not just a segment of Earth that cracked off and came back.
No, these are
Okay.
Yeah. Well, okay. So yes. So great questions, actually. So these meteors are from bits of the solar system that are further out than Earth and very unlikely to have come from within the solar system and then out to the outer solar system and then come back in again in that way. Yeah, they come from a particular population of asteroids.
All right.
Yeah. So anyway, building blocks of life came from the solar system, could be further out in space, interplanetary in origin. But what the big point here is that that Earth had the right conditions at a certain point to allow all these building blocks to potentially combine and create RNA and DNA and move forward into cells and complex life.
And when we say that too, like I always now have in my head that it's not that the conditions were once right. Conditions could still be right now. It's just every niche, as
Mhm.
Blair is often, covering every niche of the thing that is totally taking advantage of its biome and its little environment. There's no more room. Earth is full. You can't
Earth's
start
full
from
now.
scratch here anymore. There's no startup companies. It's all big block.
Except for maybe deep in the
Franchise.
Earth's core. I feel
All
like
the
that'd be fine.
instead of
Gosh, that's very crowded already too.
instead of
But
big box stores, it was building block organisms. Okay. But the life formed here on this planet. Earth Earth life is Earth life. We still don't have the evidence of panspermia, but this is potentially, this is chemical panspermia, molecular panspermia. But Justin, do you have a story?
oh, I do, but I have to just look really quick to see.
What it
Yes, this
is?
is the whole. So this is tough. This is a tough story. It has to do with death rates among children. So just bear with me if you can. So hypothetically, if I were to tell you that sharks are the leading cause of death in children, Blair would immediately react. No, it isn't. But
Slander.
yeah,
Don't
shark
slander
slander.
the sharks.
But
They
yeah,
have
you'd
it
be
hard
pretty
enough on their
astounded
own. Leave them out of this.
if I was to tell you that sharks are the leading cause of death in children. You would be astounded that it is happening that often, that it would be the leading cause, and would probably stop letting your child play unattended in the ocean. If I were to tell you that amusement rides are the leading cause of death in children, not only would you avoid them, but you would probably wonder why they still exist at all, right? If I say car accidents are the leading cause of death in children, then you likely wonder what more we could be doing to make it safer for kids riding in cars, which is a very fair thing to think about, as accidents in cars are the number 2 reason for child death. But the leading cause of child death, and I have to asterisk this, in the United States, and already you know exactly what I'm going to say, is guns. Gun violence in the United States now kills more children than any other cause, including car accidents, and pediatricians are sounding the alarm. While healthcare providers are taught to recognize and treat many public health crises, the opioid crisis, heart attacks, COVID-19 pandemic, uh, violence related to firearms is not always seen as a public health issue. Analysis of recent data, now available through the CDC, which is finally looking at gun violence in this way. Clinical researchers from the Medical University of South Carolina revealed that firearm injuries are now the leading cause of death among children under 19. They have also pointed out a large racial gap, because they're pointing out amongst Black children that has already been the case for a few decades. The article recently published in the journal Pediatrics calls for physicians and other healthcare workers to recognize that this is an epidemiological public health challenge crisis that needs to be addressed. This is Annie Andrews, MD, pediatrician, assistant professor at pediatrics at the Medical University of Southern California. She says, "When I became a pediatrician," choosing that as her the field of medicine that she would pursue, "I never thought that I would care for so many children who had been shot.
Mm.
It's not something that you think about when considering being a pediatrician or what a pediatrician does. But as a hospital-based doctor for the last 12 years, I've seen it happen again and again, and I started to get really worried about this. So, one of the things, of course, that they point to, this is basics that not everybody follows, is that secure storage, keeping guns locked and unloaded, separate from the ammunition, that can keep younger children safe from accidental gunshots if they find a gun or handling it and then it goes off. It's not loaded when they're playing with the triggers, or if it's kept in storage, that reduces chances. Also, having it locked away without having teenagers access to it. Apparently, teenage gun suicides are sometimes very impulsive actions. That, if they don't have access to the weapon, people believe it wouldn't happen. Anyway, there's an obvious need also for some violence intervention. Although I think it's a weird thing that whenever we're talking about guns in the United States with that asterisk, we have to reduce the violence in the streets and the things like this, and the culture around violence, or the bad behaviors around gun ownership. But the bottom line is, stored guns are safer than guns that aren't stored safely.
Yep.
But no guns, no guns would mean no deaths by firearms.
I don't know. Blair, tell me a story.
Yes. Well, speaking of deadly things, I wanted to tell you about scorpions.
Yes, please.
No.
So scorpions, they're actually much more deadly than I realized. So scorpions sting more than 1 million people a year. They kill more
What?
than 3,000.
Okay, well,
So-
that's not okay. First of all, that's not bad odds, but who's
That's a
the
lot.
million people who are getting? Where's
Well,
all
not
the
here in
scorpions?
California, I'll tell you.
Yeah.
But yes, there's lots of scorpions out there. There's so many. But I just don't come across them in my day-to-day. But 3,000 people die a year. So we were talking about sharks a second ago. That's like 10 people a year. So
Yeah.
scorpions is a lot more. And usually the victims... Yeah, go ahead.
Oh, and entirely avoidable without putting. If you don't put your body in the ocean, you have a 0 chance of getting bitten by a shark.
Unless,
Sure,
of course,
sure.
the sharks are raining from the sky
Right,
and they
right.
just fall on you and bite you as they fall. Right?
But this isn't about sharks. This is
Sharknado.
about
No.
scorpions.
Scorpions.
Scorpions. So usually when people get stung by a scorpion, they don't know what kind of scorpion got them. People don't know kinds of scorpions. That's not, I mean, like in certain areas you could go, that was definitely a rattlesnake that bit me. But in terms of scorpion, you're just like, it was a
Yeah.
scorpion. I don't know. There's more than one kind. And so the problem is doctors have
Which
to
I'm
guess.
just learning, because the picture that you've got up there, I guess from this article, shows a scorpion that's the size of or slightly longer than a human hand that is very bravely being set next to it. I've seen scorpions, but the ones I've seen, like, I picture them as very tiny, and we do have them in California, but they're just tiny little.
Well, we have them
tiny,
down
tiny
south
little.
in California and to the east. But if
No,
you're,
we have them in
it's-
Northern
anyway,
California too. I've
it's
seen them up there,
Definitely
but
in Northern California as well.
So, people don't usually know what exact kind of scorpion got them, so doctors have to guess. Do I use antivenom? What kind do I use? What other drugs do I use? Is this going to be deadly, or is it just an inflammation? Because it can cause everything from just pain and numbness to muscle spasms, drooling, irregular heartbeat, and even death. So, in most cases, doctors will try to do what they can to kind of treat the symptoms, but if they do decide to use antivenom, it's very costly and it has side effects. Antivenom can cause vomiting, fever, rashes, and even then, even if you do administer the antivenom, sometimes people still die if they're allergic to the scorpion. So all that to say, scorpion stings are a problem. The National University of Ireland, Galway, combined the scientific literature for data on body and claw size of scorpions related to toxicity. And they looked at about 2,500 species of scorpions. Or sorry, there are 2,500 species of scorpions. They looked at 36 species based on the available information. So a very small number of the 2,500 species. But they were all pretty unrelated as far as scorpions go. So they felt like it was a really wide swath of scorpions to pick from, I guess. And so they felt pretty confident in their findings and that it was applicable to the rest of scorpions. What they found was that the smaller the scorpion and the smaller the claws, the more deadly the venom. So they looked at these little walnut-sized Mexican scorpions, Centruroides noxious as some of the smallest, and the rock scorpion, which I think is what the picture is that Kiki just showed, Hadagenes granulatu, which is 5 times as large. And the rock scorpion, their venom is a mild irritant. That's the big guy. Whereas the venom from the tinier scorpions can straight up send victims into shock.
Wow.
And similarly, slender.
Of course, the exact opposite of what you'd be afraid of. You'd be afraid of the giant scorpion bigger than your hand. And the little cute one, you'd be like, oh, I'll hold this one. And then that's the one, of course, gets it.
Right. Well, this is also true for spiders. As a reminder, tarantulas, a lot of their venom glands are so underdeveloped that if you get bit by a tarantula, it might feel like a slight bee sting, but it's not going to hurt you. Black widows, teeny tiny, can kill you. So
have you seen, do you have black widows?
Black widows
in San
are
Francisco?
massive.
They're not teeny tiny.
They're so big.
Compared to a tarantula, they are teeny tiny. Yeah, definitely.
They're not little.
And then there's the daddy long leg spider,
which is nothing really.
Which is, but has very, very. It's a very thin, tiny spider if you think of it. But its venom is very strong, except its mouth isn't big enough.
Tiny teeth.
Yeah, I can't bite through your skin.
I can't bite you.
But so in both of these cases, the scorpions and the spiders, which by the way are closely related, this is an evolutionary trade-off. They believe that when scorpions first appeared, they relied on big crab-like claws to attack prey. But once they evolved a deadly sting, they didn't need the claws. Growing big claws, we talked about this with lobsters, I believe, in the past. Crabs? I don't remember. But evolutionarily, energetically, very expensive to maintain a huge claw. And so if it's better to stay small and you have other defenses, then you don't really need the big claws. So why does this matter to humans? If you know the size of the scorpion that stung you, that is helpful information to the doctors. If this is truly an applicable study, and the smaller the scorpion, the stronger the sting, then size can help doctors treat you when you are stung.
So I'm very curious, like, where this
Size
is that
matters.
applicable, because one of the things I would assume is, if I got bit in Northern California or Southern California, I figure there's stung. Thank you.
Mm-hmm.
There's probably a couple of possible. Where is it that they're like, you have to like, okay, what's the size of the thing? What coloration did it have?
Like everywhere it's hot. Like the tropics. Yeah.
The tropics?
Like parts of South America, parts of Africa.
This is why I don't travel.
The Middle East.
But it's body, so it's body
danger.
size, but it's also, you can also look at body size and the size of their claws, their chelicerae. Yeah.
So if you're stung by a scorpion, try to remember as much about it as you can. How big was it? What color was it? How big were their claws? This is all important. You don't need to know the genus species, but you can give as much information as possible that can help them treat you.
Whether they'll know whether or not to worry.
Yes.
When it comes down to it. All right. I have a story next that I wanted to chat about. You may have been feeling a little sniffly recently. Some people are sniffly because they have caught colds. Other people are sniffly because of allergies. But Regardless, this mucus production is a product of the immune system. It's your inflammatory response. It is the mucous membranes, the response of the body trying to get rid of foreign material. Get it out, whether it's a bacteria, a virus, a particle, something that's in the air like pollen. Your body just is like, I am snotting now. And the snot will enclose this thing I don't like and get rid of it. Well, it turns out that there are some bacteria that like to get in your body, your airways, and stimulate snot production with their toxins so that then they can eat your mucus and it makes them happy.
Jeez. Snot bacteria and eyelash mites just hanging out at the Blair buffet, huh?
That's right. That is right. Yeah. So these bacteria, we've heard of them before. Pseudomonas aeruginosa is an opportunistic pathogen. So opportunistic being it takes opportunity. It's like, oh, I'm in your nose. What do I get to do? Woohoo! And it produces a toxin called LAS-B, and this causes an epithelial response where the epithelium produces a compound called amphiregulin, and that causes type 2 inflammation and mucin or snot production. And so the researchers in this paper that is currently published in Immunity, they're calling this niche remodeling. So basically, this bacteria is promoting colonization of your tissues by producing this toxin, which makes the snot, which they can eat. And then they're like, more of us. And then there's more bacteria and more snot, and it's better for the bacteria. And it creates allergic sensitization. And so these bacteria may actually be stimulating your immune response and making you more highly responsive to other allergens as well, because they've just started that immune response. So it's an interesting I guess behavior, or it's 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 opportunity, right? This bacteria is like, ooh, snot, I like it, I'm going to eat it, it makes me happy, and it just so happens that you produce it when I secrete this toxin.
DNA is food.
Everything.
So how do I know if this is why I'm snotting, and how do I get rid of them?
Right. Well, that's
That's a
the
different
question,
show.
right? That's a different scientific answer, right? I
That's
mean,
an advice show.
there are the things that you can do constantly, like a neti pot or
No, but be careful because those
nasal
will kill
Distilled
you.
sprays.
water. Yeah,
Those will
If
distilled.
you're
also kill. Yeah.
It has to be clean water
Yes.
that you have sanitized. Yes.
Yeah. Don't trade the
Or these
bacteria
nasal
for a meat
And
sprays
it
that are
has to not
available.
still be hot.
Yeah.
Like if you boil the water, it has to cool down first.
Not
I learned
boiling
that the
water
hard
in
way.
your nasal cavity. No, that's going to be a problem also. Be careful how you use the neti pot. Yeah, but there are techniques, especially during allergy season or year round, if that's the kind of thing that disturbs you, where you
Oh, that's a
can
good. Stick honey up
rinse.
your nose.
Honey is antibacterial. I don't know if that's a good idea
I
necessarily.
think.
Maybe
I think.
medical grade honey that's been diluted with with distilled water, and I
You're gonna
don't know.
wake
I
up
just don't
covered
think
in
I'm not
ants.
medical. I'm not giving you medical advice right now. I'm just telling you about this new thing they found.
Oh man.
Oh god, as like one of those medical advice shows where they're like 3 cures for that. We would do such a terrible job.
We would do an awful job.
We just start throwing stuff out there, just spitballing and speculating. Yeah, what if you stuck honey in your nose?
Why
Yeah,
did I
just
do this? I don't know.
poison the ants.
All right.
Yeah,
Well,
and
what if
scorpions.
we
So
needed to cure? What if we needed to cure our liver? What should we do
that's
then?
a great question. Ooh, well, or just about anything else if we wanted to stay young forever, as Blair is so gracefully doing so far. We will need our Yamanaka factors. Yamanaka factors are 4, count them 1, 2, 3, 4, cellular reprogramming molecules, which have been shown to slow the aging process and improve muscle tissue regeneration capacity in mice. But these are already known. Salk scientists used these Yamanaka factors to see if they could increase liver size and improve liver function while extending health spans of the mice. The process involves partially converting mature liver cells back to younger states to promote cell growth. Results of this study are published in Cell Reports and reveal that the use of reprogramming molecules can, in fact, improve cell growth and lead to better tissue regeneration in the livers of mice. Quotey voice. We are excited to make strides at repairing cells of damaged livers because someday approaches like this could be extended to replacing the whole organ itself, says one of the authors, Juan Carlos Ispizua Belmonte, professor at Salk's Gene Expression Laboratory. Our findings could lead to the development of new therapies for infection, cancer, genetic liver disease, as well as metabolic diseases like non-alcoholic steatohepatitis. So kind of one of the interesting things in this story too is the way they went about this, because people have been trying to use these factors, these molecules, to get generation going on tissues before. One of the problems, though, is it sort of becomes a stimulant for cellular growth. Works great, but it doesn't come with the instructions of regulation. So you can have tumor formation. You can have overgrowth. You can actually create a sort of form of cancer, or at least benign tumors that are cancer-like. So you can get this little bit unrestricted growth. What they did is they managed to find a nice balance there, giving a small amount over, I think, just 1 day's worth of treatment. And this is Toma Oki Hishida, who's associate professor at Wakayama Medical University in Japan, but who was working in this project as a postdoc. Yamanaka factors are truly a double-edged sword. On the one hand, they have the potential to enhance liver regeneration in damaged tissues, but the downside is that they cause tumors. We were excited to find that our short-term induction protocol has the good effects without the bad: improved regeneration and no cancer. Also in this, they found that there was a gene, TOP2A, that wasn't known before to be involved, a mechanism involved in the reprogramming. And it was highly active immediately after they did the treatment. So it's an enzyme, topoisomerase 2A. It's an enzyme that helps break up and rejoin DNA strands. So they noticed that it was upregulated, so they went and blocked it. They did another experiment where they had blocked that gene, and they saw a 40-fold reduction in cellular reprogramming rates, leading to far fewer younger cells. So not only have they found an application or a protocol to use to stimulate regrowth of these livers that doesn't have the negative effects, they may have found another mechanism that is highly involved on the protein level in doing proper regeneration. But yeah, these are these sort of little steps along the way to science created evolution. Where humans may one day be like, oh, I gotta go by the doctor. Oh, what's going on? I gotta get my liver regenerated.
Right. Well, I mean, our liver is so regenerative as it is.
Already,
Like
right? Yeah.
you can have a little tiny bit of liver and it'll like grow back. Our liver is amazing.
Yeah.
But at the same time, once you really damage it, the hepatitis and other aspects it really is it you need liver transplants, and people get those occasionally. This whole thing though, it's fascinating to me. Like, number one, as this gets developed, what is going to be the trade-off of like how bad off you are for your liver damage before you're allowed to take this regenerative cocktail to regenerate your liver? Or secondarily, now that they have this protein, is this going to be a target that could be like, oh, well, I have a slight dysfunction in my liver because I drink every weekend too much. And now I can just pop a pill after have too many drinks and suddenly my liver's healthy. Like, so, I'm just wondering what the real clinical application or even just over the counter, what are we going to be getting eventually? Be
Yeah.
fantastic. I've thought it would be fantastic to have liver regeneration for years.
Mm.
This is
It's
great.
liver, but and they worked on that. And when I was reading this, I sort of thought, well, that's a soft target. That's something that can already do some level of regeneration. But what I take away is like the fascinating part about this is these are things that are already taking place in the body on small
Yes.
scale.
Small scale. Yep.
Small scale, like, let's go repair these 100 cells over here. We need to regenerate this, fix this DNA and this one bad cell, whatever it is. On a very small scale, the body has the ability to do all sorts of amazing repair stuff. But what we're doing then is saying, okay, we
Now let's do
like
it
that.
bigger scale.
Let's do it again and again, 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 again,
Yeah,
again.
yeah,
Yeah,
yeah.
let's do it
Let's
really
go.
big. And so we're not so much science isn't so much inventing a thing that the body can't do on its own, but giving it access to do it on like you said, on the bigger scale.
Bigger. That's what people want. We're always like, we want more and faster and cheaper. Let's do it. And another really fantastic discovery of a new target. Researchers have just published their work in Nature on discovering a mutation that's responsible for extreme lupus. So, studying a young girl back in 2016, a 7-year-old Spanish girl named Gabriela had
Oh, no.
symptoms of severe lupus. And so they coded her, they sequenced, decoded her genome, and they found a single base variation. And it was a change in the gene for TLR7. TLR7 has never been implicated in lupus before. And it's a really important gene because it might be the central signaling pathway in lupus. They say that TLR7, it's otherwise known as toll like receptor 7. It is a pathogen detecting protein. And so normally it's exposed on the surface of immune cells, bumps into pathogens, and says, you're a pathogen, and tells the immune system to attack a pathogen. But in this case, with this mutation, it it misidentifies cells within the body, and so it creates the issues of autoimmune disease. And in this particular case, because they've now discovered this particular mutation, they altered the gene in mice. And they had the mice start expressing lupus like symptoms. And they also found this mutation in several other lupus patients when they went to look for it. So now it's a target, and there's proof that this is, probably not the only mutation, but a very important mutation in the cause of lupus and potentially other autoimmune diseases like lupus. It also explains why most people who get lupus are women, because TLR7 is on the X chromosome. And this is where both copies of the gene are often, they get expressed. So when men get just one copy, they won't have the expression of the disease phenotype. So now, who knows? It's a path. It's a link in the chain to the pathway that could lead to better treatments for people with lupus. Yeah,
And
super
that's just
cool, right?
one of the one little jeez.
Yeah, one
Some genes are
single nucleotide mutation, like just
yikes.
one little switch in a gene. Yeah. But I think it's this kind of work. This is the first time they've really discovered something this big. And so it's a huge discovery for the field, and maybe And it's interesting. We've talked, the other aspect to this is that we talk about gain of function research in how we, in our genetic research, when we try and figure out how viruses work or how to find treatments for viruses, we'll often kind of give proteins additional functions. So, this gain of function by making changes in them. This TLR7 mutation is a natural gain of function variation. This is like a switch that is a gain of function process that happens naturally within the body. So I don't know, just an interesting little connection right there. And maybe we will find more cures, more treatments, more targets when we look at these things down the road. This is This Week in Science. Thank you so much for joining us for the show tonight. If you're enjoying the show, please tell a friend. All right, Justin, let's come back for this COVID update. What do you have to tell us?
Bum, bum, bum. Well, these are look backs. This isn't current stuff going on with the pandemic that we're in. This is a look back at the pandemic of yesterday.
That we're in.
Last year. Last couple years. And we're still in, but we're looking at this is a couple of studies. I think it's April 2020 to June 2021 is the time frame. And it looked at eliminators versus mitigators. Classic struggle between eliminator and mitigator strategies of countering COVID-19. It says here the pandemic policy measures implemented by countries that tried to control rather than eliminate COVID-19 are associated with slightly worse mental health and lower life evaluations, according to these studies. And this is published, both are published, I think, in the Lancet Public Health Journal. Countries that aim to eliminate community transmission within their borders, those are the eliminators, experienced fewer deaths and equivalent or better mental health trends during the pandemic than countries that aimed to control rather than eliminate the transmission, mitigators. Governments around the world employed diverse strategies, issued a variety of guidelines. So they had to use 15 countries, but also knowing that there's variation in how implementation took place amongst those 15 countries. So some countries really tried to eliminate down to a zero community transmission. Other countries slowed it down we had some lockdowns, there was some workplace business, school closings, social distancing, wearing of face masks, cancellation of public gatherings and public transport. That was really sort of more of the mitigations though. Right? So it says how this they weren't focused on what were just the health outcomes. We wanted to know all of the mental health outcomes of these as much as possible. So countries that were eliminators were Australia, Japan, Singapore, South Korea. Mitigators, Canada, Denmark, Finland, France, Germany, Italy, Netherlands, Norway, Spain, Sweden, and the UK. The US was not part of the study, but definitely would have been in the mitigator camp.
Yeah.
It's pretty interesting, the findings. Basic findings here. Mental health of people in countries that tried to mitigate the virus transmission were more impacted than people who tried to eliminate. So if you went to a hard lockdown, we're going to get rid of travel, nobody go to work, stay home, we're going to time out. People had better health outcomes, mental health outcomes as a result of that. Because they partly say, because it worked. And people got to go back to more of a life as normal, versus the mitigating countries had such a long drawn out, even though they weren't as restricted. They had to do all this social distancing and elimination of events over time. Everything was just drawn out.
I have a theory. Mm-hmm. Perhaps it's specifically that if you were completely locked down, you had no choice. But if you were in the mitigation phase, you had to make your own health decisions. every day in everything that you did. And the
Right.
mental
That's a lot.
strain
Yeah,
of
huge
that,
mental load.
it's constant anxiety and stress and
Mhm.
guilt
Right.
the whole
Yeah.
time. This is why
Yes.
in 2020, the first half of the lockdown of 2020, I was actually happier with than the second half, because in the first half, you could do anything. And there was no guilt to not doing anything, and there was no guilt to doing what you had to do to get groceries every 2 or 3 weeks. But the second half, you were suddenly allowed to do some things.
Hmm.
You're
So
like, has it
that's
been long enough since I did the last thing? Is this one safe? Yeah.
Yeah, that's very observant because the length of lockdown and like everything closures had no impact on mental health reporting. Like that's what we're all doing. No, like you say, guilting, no decision making, no worry and stress over the different interactions. It didn't matter. The lockdown was 6 weeks or 3 months. It didn't increase in negative mental health. But the with some exceptions. So there's some interesting outliers in this. Let's see. See if I can find this here. Analysis found that lockdown had a significant, but relatively small adverse mental health effects, while the experience of lockdown slightly lowered mental health scores across the study population. Females were more likely to suffer mental health consequences than males, especially those between 20 and 29 years of age. There were no significant effects for adolescents of either gender, no effects for younger males, 20 to 29. Meanwhile, males aged 55 years and above saw an improvement in their mental health scores during lockdown. Only
Interesting.
demographic.
Their
That,
family had to be with them.
They finally didn't have to
or
go to work. They're
or
like, I'm
well, no,
ready.
this
This
is
is
also
great.
this is 55 and above. Maybe it's that. Maybe it's that. Like, oh, I don't have to interact with anybody.
Uh-huh.
Whew.
I can stay home, work on my model painting.
Yeah. So moderately large effects were also found for females living in coupled households with dependent children. They were more likely than any other group to face negative mental health outcomes. Were males in coupled households with dependent children or without children saw only modest negative outcomes.
Mhm.
So women in that situation having worse mental health outcomes. Interestingly, again, there was no negative effect of lockdown found amongst single mothers.
Hmm.
Which, what does that tell you? Women are stressed out by their partners.
No.
They
I ha-
were already doing it all, I think,
Yeah.
is probably what that meant. And they suddenly couldn't go to work, right? So you actually
Yeah.
got to see your child. If you're a single mother, you're probably going to work at least 40 hours a week.
Yeah. Or
But
maybe you're a single mother and you're now in lockdown. You've decided to go be in lockdown with a family, with your whole family, and you're not going to work, and so you've got help with your child. Everybody's taking responsibility. There are
yeah,
also so many
that's
interesting
helpful.
aspects here.
I
Yeah.
just like the idea that confirms a thing I've always thought that men and women who live together, it's much harder on the woman.
Well, it's early on in the pandemic, I think we reported on a study that women were taking on the majority of childcare duties
Yeah.
while teleworking versus
Yeah.
men. So it's, they were expected to keep the clean house and take care of the children and cook the food and do all the things that we thought we left in the dust in the 1950s. But here it is again. They called it like, what did they call it? But it was like the new 1950s, the first 6 months
Yeah.
of the pandemic, when all of a sudden women were
Interesting.
expected to do all of these things in addition to working from home.
So, but
Yeah.
anyway, in a nutshell, the Justin plan, which we all know is a total lockdown of everything for 6 weeks, where nobody goes out of the house and just waits until it's safe to go outside for the virus to be gone, would have actually had much less negative, much more positive mental health outcomes, let
Right.
alone the health outcomes, than even the strictest things employed elsewhere, except maybe China. Maybe I'm an authoritarian.
Yeah.
Maybe I think
There was
it's actually
another
good.
study that came out, I thought, that showed, uh what was it? There's something that came out. Oh, I think it's actually your next story, based on what would have happened in terms of deaths, not just,
So yeah,
yeah.
there's a few of them out there. Another one's like the actually the less restrictive, the ones that like the one that's the mitigator one actually created more governmental distrust in countries than the ones that were like, oh, we're going to lock down and wait till it goes past. And then the one you're talking about, they sort of looked at a third strategy. It's not really the mitigator strategy. It is a mitigation strategy, I guess,
It is,
but
yeah.
it's a very specific one where they this idea that you could just focus on the most vulnerable in society, the elderly, the already sick, and isolate them in society, and let everybody else continue to
Live our
intermingle
lives,
and
yeah.
live our lives and get that herd immunity. And basically, they just did a bunch of modeling. They used, I think, London as the example, but they made it a population of 1 million in their modeling, and they ran all these scenarios. Basically, what they decided was
Terrible.
even in the most perfect version of shielding the vulnerable, which they say also is impossible. Everything is leaky.
Yeah.
Humans, wherever
People are
you are in
leaky.
that low risk, high risk, whatever. Yeah, everything leaks. But even if you did the perfect one, you would be overwhelming hospitals and in that million person model, leading to tens of thousands more deaths. This shielding or focused protection. It's published in Public Library of Science Global Public Health. This is University of Bath study. Said no, no, that a lot of people have thrown that out there as a thing we should have done, but based on their modeling, that would have been disastrous.
And some countries did try to do that, and they did have very high death rates. And if we had used those, yeah, there were a couple of studies early on in the pandemic that were proactively trying to figure out what the best response to the pandemic would be in various countries, and that they predicted all sorts of deaths and economic losses as a result of not locking down and not doing as much mitigation as possible.
And
So
I
we're
feel like we've
seeing it.
surpassed them all.
Yeah, we've surpassed all. Oh, good job, everybody.
Yeah.
But as we move forward, we've got vaccines now, and we have all sorts of other treatments. And it turns out that there is a new mRNA vaccine potentially on the block. Researchers, or not researchers, a vaccine company, Arcturus Therapeutics of San Diego, did a placebo controlled trial of its candidate mRNA vaccine in more than 17,000 participants in Vietnam. It reported that the vaccine had 55% efficacy against symptomatic COVID 19 and 95% efficacy against severe illness and death. So, was was working on the
That's
same level as the other mRNA
Wait
vaccines,
a second.
basically at the same
Is
level
it?
of
That sounds.
efficacy. Mhm.
That does not sound as maybe the vaccines
Against
work.
Delta and Omicron. Yeah, because of the newer variant, and that's what it was up against. The interesting
Wait,
thing about this mRNA vaccine, though, is that it's not exactly like the others in that it is amplifying. So, the this particular formulation is different from Moderna's and Pfizer BioNTech's vaccines. Those have mRNA that codes for the SARS-CoV-2 spike protein, and then it gets into the cells and uses the cellular machinery to create the antibodies to the spike protein. So this happens also with this self-amplifying mRNA, but the self-amplifying mRNA has a little bit of code in it that says, we're going to copy ourselves. And so once it gets into the cell, its little particles open up and its copying instructions start copying. And so it superpowers the amount of mRNA that ends up in your cells so that the mRNA lasts longer, so that your body has a longer amount of time to create antibodies. So, number one, you can have a lower dose vaccine. So, each shot is a lower dose. Secondarily, their formulation is freeze dried and can be stored at room temperature. So you don't need to have the special refrigeration to enable transport of this vaccine. So there's still a lot of questions, and it has to go
That's
through.
very important. That
Yeah.
was you can't trust refrigeration
Well,
at
plus,
this
when
point.
somebody asks you which
Everywhere in the
vaccine
world.
you got, you get to say Arcturus.
Arcturus, the
Yeah.
bear.
So I'm still confused. Maybe I've been overly optimistic about my interpretation of numbers of efficacy and things. I thought the mRNA vaccines that we had now were like 90 percentage is effective of
Against
preventing
severe disease and death.
just from that. Just you could still 50-50 chance of getting it, even with the
Yes.
oh
Yes.
Well,
gosh,
think
that's not
about
good enough.
Yes.
I heard on NPR this morning that 60% of uh the entire US has gotten COVID. 60%. And so based on that number, we know there's a very good number of people who've been vaccinated, so that would have to be the case.
Yeah.
I'd hope so. Yeah.
But I just figured that was
But
like
being
ran
vaccinated,
down.
yeah, being vaccinated is the thing that will give you better protection against future variants of the virus. Um, being just infected without being vaccinated, you have a higher probability of of getting really sick and dying, and then also getting sick again because you're not going to be as protected against future variants potentially.
Omicron.
Yeah, future Omicrons. Omicrons and their babies, the baby Omicrons. And then finally, there's a retraction that we should all be aware of. Earlier in the pandemic, it was highly reported, this paper, that high doses of vitamin D are beneficial to people with COVID-19. It was published in Scientific Reports. It has been retracted now as researchers reported that patients were not appropriately randomized, and so the differences in the
Uh-oh.
outcomes couldn't be attributed to vitamin D therapy after all. So back to the drawing board on that one, but it was a
So
highly, highly cited paper. It's been
I'm
retracted.
very tempted to say like, yeah, that didn't make sense. I didn't believe that in the beginning. But then there was somebody who could probably play me. I'm like, this is great news. Vitamin D's. I don't remember what my take on it was at first. But when I heard this one, I'm like, yeah, I don't know who this vitamin D industry. Like, there's got to be somebody. I don't even know how you get vitamin D, the industrial, like when you're getting supplements or when they've added, put it into the milk or where this comes from exactly. There's got to be some big money in that because I feel like there's always a study in the peripheral saying how wonderful vitamin D is. Just it's like more you hear about it more than the other vitamins, so I kind of feel like there might be an industry push to make people take vitamin D.
I don't know how much I don't know. I have no idea how much money's
Everybody
in the
here in
vitamin
Denmark
D
takes it just
supplement. And
because
I mean,
there's
supplements are money, but
very little
whatever.
sunlight
Yeah.
getting in.
This is This Week in Science. We hope that we are a ray of sunshine, a ray of science sunshine into your curious world. If you do enjoy the show, please head over to twist.org and click on our Patreon link and help produce the show. We rely on your donations to keep the show going week after week, keep the equipment up, and keep our connections through the interwebs and all these things. So, very appreciate your support. $10 and more per month, not even per week, per month. And we will thank you by name at the end of the show. Thank you for your support. We really can't do this without you. Okay, it is time. If Blair's internet's working after, all these days and weeks, it's time for Blair's Animal Corner. Da-da-da, with
With Blair.
Blair.
She loves our creatures, great and small. Biped, millipede, no pet at all. If you want to hear about animals, she's your girl. Except for giant pandas and squirrels. There's animal
What you got, Blair?
So I want to talk to you about catapulting spiders. I'm very excited about it. So these are Philopenella prominens, hackled orb weavers. And they, like many orb weavers, well, if depending on if you're a male or female, have a very interesting methodology to mating. So the females like to try to eat the males afterwards. The males want to survive. But they still have to mate because, that's how you win life, is by spreading your genes. So it's a tough thing. How do you do that without getting eaten? And so what researchers of Hubei University in Wuhan, China found is that these spiders will catapult away from the females as soon as they are done mating. They fling themselves away from their partners at up to 88 centimeters per second. And I did do the math, and that's about 2 miles per hour, which is pretty
Quick
fast.
for a spider.
Yeah. For a little spider coming from a dead stop, for sure. And so what they found, looking at 300 individuals, 155 individuals, successful matings. So actually it's about 310 individuals in 155 couples. They all of the males that survived catapulted away to do so. All the 3 males that didn't, they were all eaten. And then to test the study, they prevented 30 of them from catapulting away. I don't know how. And all 30 were eaten. So that catapulting was essential to surviving reproduction.
Oh my gosh. So there's the catapulting, you could also say maybe slingshotting themselves, just getting away as quickly as possible.
Yes. They accelerated an average about 200 meters per second squared. And
That's
as they
quick.
soared through the air, they spun 175 times per second. Yeah, and so what they found was that they achieved this by folding their tibia metatarsus joint against the female, and when released, the way that spider legs actually hydraulic pressure. So the hydraulic pressure, crazy because since they don't have bones inside or muscles, it's all pressure based stuff.
Right.
So it's hydraulics. Yeah. So and this is these oh God, what are they called? Hackled orb weavers. They actually live. The reason I said 300 before is they live in groups of up to 300 individuals in a web complex, and so there's a lot of kind of negotiating and interconnected social situations that happen in these spiders. They are social spiders. And so beyond that, it could also count as a sort of quality control for females. They don't know this yet. This is something they want to study next. But is this a way the females are going, oh, you didn't get away from me fast enough. I'm not going to use your sperm. I'm going to go mate with somebody else. So it could be an extra selection technique from the females as well. Or the females just want to eat because they just expended a bunch of energy mating and they want a good energy boost for the babies. I don't know. Could be either.
The females definitely want an energy boost for the babies. I mean,
Yeah.
that's going to be important for, nutrients to support the eggs and to make sure that they have a chance at survival. I mean, I thought that was part of why they want to eat the males to begin
Yes.
with. Give me my nourishment for my babies.
That's right.
Tell us
Next.
another one. Yeah.
Yes. You want to hear about a 75-year-old great tit study?
Yes. I love the
Yes.
great tits.
Yes. So this is, as far as we know, this is the longest study in oh gosh, I have to find the exact wordings. The longest continuous study of an individually marked animal population in the world. This is a 75-year-long study of great tits in Oxford's Living Laboratory at Wytham Woods. So it's special because it's not inside a lab, it's out in the wild. They've been able to very specifically study these great tits, individually marking the animals. And so through this, they've been able to learn so much about these birds. They've been able to release a bunch of studies. But the one that this particular study, or this particular breaking news from this week is about the fact that when the study started in 1947, the first egg hatched on April 27th. This year, in 2022, 75 years later, the first great tit egg hatched March 28th. So it's a whole month earlier.
Yeah, we've talked before about birds being, a week or 2 weeks different as a result of climate changes and
Yes.
temperature differences, but a month. That's a
And-
lot.
Yeah.
And
And I'm
so-
sorry, I
Blair-
said the wrong word. Not hatched, counted. The first egg was
Okay.
counted. So the
Okay.
first egg was laid-ish based on their
Yeah.
daily checks, right? So it's still
So-
very important. Yes.
So Blair,
Yes.
what would be the implications? Why should we be concerned that the egg shows up a month early?
Right. So, good question. Hopefully, this study will continue for another 75 years and we can figure out exactly what the impacts are. Now, what we do know about great tits from the last 75 years of the study is that the timing of when they lay their eggs is influenced by many things. Climate, the big one, social interactions, health and behavior of trees. And so all these things together relate to the timing of when they lay their eggs. Now, if the climate tells them one thing and the tree is telling them something else, this could be deadly. If the climate is telling them one thing and the other tits are acting differently, that could be a problem. If they're acting one way and bugs aren't coming out yet because it's not warm enough yet. There's all sorts of things that can happen if you get kind of asynchronized with your normal schedule. The tits were the perfect subject for this long-term study because they take well to nest boxes. They breed at high density, so you don't have to walk all over the place checking on your birds. They don't travel far from where they're born, so you get a kind of consistent cohort over many generations, and they cope well with being monitored by scientists. They can get grabbed and very delicately selected and they can be tagged with little leg rings and it won't impact their breeding or their health in other ways. For other birds, they're very sensitive, and if you do that, that can mess up their whole year or life. So this is a situation where they're a perfect study for that. And so they looked at breeding data from 1,209 fixed location nest boxes. That's a lot of locations over many years. And a study this duration is built on hundreds of people, at least tens, if not hundreds of publications. And so this has been something that's been kind of handed through many generations, and it will continue, hopefully, to be handed through because it's important for us to assess the changes that climate change are pushing onto animal species, because not just are the tits affected, but what did those birds affect around them in the ecosystem? This is all really important. So climate change can have effects on all sorts of biological systems. And in this example, birds are adjusting their behavior and reproductive decisions, but that can also change the circumstances of other living things around them. So the next step of research is to combine measures of timing of the trees with data on insects and apply those to the birds. So that's exactly what you were talking about, Justin, is how are the trees and the insects reacting? Are they reacting at the same rate to climate change as the birds? Spoiler, probably not. And so how does that impact everybody? But a
Yeah.
whole month. Can you imagine a whole month difference? That's huge.
It's massive.
Mhm.
And I question whether or not they are on track with the insects and the plants and the other things. It's hard to imagine that they and other species are keeping synchronized. Things are
Well,
probably becoming unsynchronized as we move forward.
yeah, and that would be the fear, right?
Yeah.
But I think that then there is some I think there's some logic in the way that world exists, and the way that the things within the nature tag the climate for their interactions, tagging the climate for when the tree is going to blossom, tagging climate for temperatures being when insects will breed or when the birds will start laying eggs. If they're synchronizing to the same touchstone, if they're synchronizing to the same North Star of climate, then we should be okay.
Right, but the problem is how adaptable and how flexible are those
Yeah,
timelines?
yeah. How
That's the
quickly? How
problem.
quickly that takes
Different
place?
And
different
and
types
the
of
touchstone,
organisms can
yeah.
respond differently.
Yeah, which maybe they're all responding to climate change and the change and the changes that are happening, but maybe they are responding at different rates. Maybe they are responding differently. And so you're going to see it's not going to match up. It's not going to be this beautiful everything stepping in time. It's going to be messy because that's biology, and this is not it's not a terrarium that you're,
Mhm.
fixing and making it work. It's
Yeah,
Yeah,
in the
except
case of
oops, it's the terrarium I left in the hot window in the kitchen on a summer day.
except that I think I would
Across the
imagine
water.
that there is enough of a rogue winter that goes longer than it normally does. It's not going to. I guess I don't think you can see a 1 year collapse of a
No, it's
dramatically different weather
multi-year because of
pattern.
the trends, and that's what we're seeing is that over years and
yeah,
years,
75
there are
years
you
has
have
changed,
you have
yeah.
Yeah,
boosts in
yeah, yeah.
populations, you have reductions in populations, but over time, because of the change, you're seeing species not able to keep up with the rapidity of the change in climate, and that's the problem, is that climate is changing too quickly.
Mhm. It's the thing that I often think about is when randomly you get a hot spell in January and then a bunch of trees flower all at once and release pollen in January, like it's
Yeah.
April, you go, was that your whole shot for the year?
Hope not. There's
Because
some more
you're
frost
in
coming.
trouble then.
Yeah, or it's we just had a bunch of snow, very late snow here
Mhm.
in the Pacific Northwest, and it froze a whole bunch
Mhm.
of cherry blossoms, plants that had started their whole spring thing. They got
Uh-huh.
it
They're
because
out
it
of
was
luck
spring and they were ready
this
to
year.
go, and then the late winter hit. But I wanted to add that the last thing for this is that as the climate is changing very quickly, the climate doesn't care about species in particular. And so some species are going to adapt and do great, and they're
Mhm.
generalists and they're going to find new food sources, and maybe they'll even do better. But other species are not, and they're going to just be like Blair said earlier, I can't handle it,
Yeah.
and they're going to die. And that is how it marches forward.
And that will have huge impacts on other animals,
Yeah.
plants, biomes, and
The web.
humans, depending. So yeah.
What about bloodworms?
Oh, yes. Let's talk
Let's
about bloodworms.
Yeah.
Did you know that bloodworms have fang like jaws made of protein, melanin, and copper?
No, I wasn't really aware of bloodworms either, so
Copper
this is a whole thing.
fangs.
Copper fangs. The concentration of copper in their jaws is not found elsewhere in the animal kingdom.
So
it
people
has
should be stealing copper from them instead of the
the
copper
catalytic
wiring from
converters and things. Yeah. So it yes. So the confusion here and the thing that scientists have had a really hard time figuring out is, where do they get this copper from? How do they synthesize it? How do they turn it into teeth? Like, it's just nothing that there is a precedent for in the animal
I'm
kingdom.
just really concerned about worms with these sharp teeth. This, I mean, it's like a toothed sandworm, except it's after blood.
I like the idea
Yes.
that the first to reach copper technology on the planet is a bloodworm.
Yes. So bloodworms, they only make their jaws once in their life. It's not like they lose teeth over time or anything like that. They need to be strong and tough enough to last for their entire lifetime, which is 5 years. They bite prey. They puncture straight through an exoskeleton sometimes, and they
Whoa.
inject venom that paralyzes their victims.
They have venom and copper fangs?
Yes.
Wow.
Yes. The co-author of this study, Herbert Waite, said, quote, "They are very disagreeable worms and that they are ill-tempered and easily provoked." This is from
cranky worms with teeth and venom. Oh, this is getting better and better.
Yes, this is from UC Santa Barbara. And Herbert Waite continues to say, when they encounter another worm, they usually fight using their copper jaws as weapons. So what they found was that they are actually pulling copper from marine sediments to form their jaws. They begin with a protein precursor, which recruits copper to concentrate itself into a viscous protein-rich liquid that is high in copper, and phase separates from water. The protein uses the copper to catalyze the conversion of the amino acid derivative into melanin. And then they then combine with protein, giving the jaw mechanical properties that resemble manufactured metals.
Yeah, biological metallurgy. Wow.
Yes. So this worm is able to
worms.
Yeah, they're able to synthesize a material that you can't really do in a lab. In order to do it in a lab, I mean, you could, but it would be extremely complicated. It would involve different procedures, apparatus, solvents, temperatures to get to this point. It is a complicated thing to do in a lab. But this worm is just pulling copper straight out of the sediments, making teeth. So the hope is that this will provide a better understanding of how bloodworms conduct this processing to streamline parts production for industry. Of course, it always comes back to how can you synthesize the copper like the bloodworm does.
Yeah, exactly. Can we take that methodology or that process and simplify it or put it into a manufacturing system? Can we make that work?
But in the meantime, also just, venom injecting teeth made out of copper from marine sediments in a bloodworm. What?
That's insane.
Yeah.
Cranky bloodworms.
Oh my.
I gotta look up because I want to avoid
bloodworms.
Yeah, where are these things?
Oh wait, fish food. Are those the little tiny worms that you get in the frozen blocks that you put in the aquarium and your fish go crazy?
Yeah, those are bloodworms. Yep.
And they have these little copper teeth. I did not put that together.
I don't think they can bite through human skin, I would guess, but
no, they're little tiny things. Oh my goodness.
Yeah. Wow. Although another study, now I'm just Googling bloodworm, and it does say
everywhere
that sometimes the venom can cause a reaction similar to a bee sting, depending on the species.
So the big, maybe, I don't know if the size relationship holds for worms the way it does for scorpions and spiders. Little tiny bloodworms with copper teeth might be more dangerous.
Now I'm looking. Is that the same thing that you put in an aquarium? So yeah. So the
according
Glyceridae
to Dickie
family.
In the chat room, yeah, they'll always be fish food to me. Yeah. So it sounds like it might be the same.
Yeah. Where's their genus in this study? Yeah. So it's a large genus, I think. So I would guess that the ones we feed to fish are small. Yeah. Glycera dibranchiata is what this study was on. I'll
Yeah.
bring it to the after show. Well, I'll do a little bit more bloodworm
Let's
research and
yeah,
we can chat
let's
about it.
dig into bloodworms in the after show.
Yeah.
Sounds like a
They're
tasty
annelids. This is all I
aperitif.
know so far.
Digestive? Digestif. Hey, Justin.
Yeah. That's I
Yeah, do you
know
want
that
to
guy.
talk about fire? Are you ready to talk about things
Oh
that
yeah. So
burn?
there's a couple of fire stories this week that uh I picked up just before the show started. One is actually sort of a continuation of a study I talked about. It's a different study, but it's a continuation of a theme that I've been talking about, which is indigenous stewardship of lands. We were talking a little while back about a population of caribou that an indigenous population has been resuscitating and getting its numbers up. This is a University of British Columbia study that looked at it controlled fires, controlled burns, practices of indigenous peoples, and saying that Canada needs to rely more on those traditional burning activities to sustain the environment. This is actually a quote from Dr. Amy Cardinal Christensen, uh, Indigenous fire research scientist at Canadian Forest Service. Cultural fire is using fire on the landscape to achieve certain cultural objectives, such as sustaining diverse animal life and plants that serve as medicine or food, according to Amy Cardinal Christensen. For example, certain berries tend to fruit prolifically following a fire. Cultural fires are typically low intensity, small scale burns that are community driven and practiced by indigenous firekeepers around the globe. I got once asked who has better knowledge of biology, a scientist who has studied biology with a PhD, or an indigenous person who's been living in this same biome for multi generations. And it's a tough question because one is you think, well, one guy that understands our scientific shared knowledge of mechanism about how certain things work. And the other one
Right.
actually just knows that this bird eats this fruit, and that if you, like in this case, if you do this controlled burn, that fruit will grow more, then there's the birds are going to be there in greater population, whatever the thing is. So there is this scientific knowledge, but there's also a cultural knowledge that can we can all be benefiting from. And that's basically what the authors of this study were saying. They're looking to knock down what barriers the not British, the uh, Crown territories, which is what the Canada refers to these as, that the indigenous people land adjacent to the indigenous lands that still exist in Canada. On the indigenous lands, controlled burnings are taking place and they're being successful. The adjacent lands that are controlled by the Canadian government are not having the same results because they're not being managed in the way that traditionally those lands have been managed for tens of thousands of years. So they're making a push to knock down those barriers to allow the indigenous groups to take over more land, which again is a theme that we've talked about a bit on this show. If you want to preserve lands, if you want to preserve it, give it to the humans. Just turn that land over to the humans who've been doing that successfully for tens of thousands of years
Right.
and have a vested interest in the success of animal populations, of the environmental health, and that sort of thing.
Well, I
So
think also when you start thinking about it as the vested interest, it's not just necessarily a business interest where it's one company who wants one particular kind of wood in a tree for, for their the wood for their logging that they do for selling wood for houses. There's much more resource use that's involved, and the cultural aspects are they're deeper, and so there's a lot more interest to maintaining the forest in a way that is sustainable for
Yeah,
everyone
and I mean,
involved.
even if it's not being, even if the resources aren't being used for something like logging, like you're saying, which can devastate
Yeah.
a biome. Uh, the hands out, like we made it illegal in California for indigenous tribes to do their controlled burning. And what do we have now? Massive wildfires. Same
Yeah.
thing in Oregon.
Yep.
Made it illegal, massive wildfires. All these, and so there's all these benefits that, how long have people been, in California, for the federal government really been in control of California? And I'd say it's maybe 100 years of actual, like, having policies and involvement and that sort of thing. It's very new to a territory to try to have understood how all these interactions, how all the interplay goes on in a biome. And
Yeah.
especially since most of it is not done by scientists in the first place, but by politicians and bureaucracies and the rest of it. So
Yeah.
yeah, turn
I think
lands over. You
the
want to protect
framing
land, turn it
shift
back to
is
the.
yeah, the framing shift from prescribed to cultural, I think, is just interesting in itself. That's gonna have room for study for years.
Yeah. And then the last study I've got here is, this is UC Riverside researchers showing that fungi and bacteria that are able to survive redwood forest megafires are microbial cousins that often increase in abundance after feeling the flames. Fires of unprecedented size and intensity, called megafires, are becoming increasingly common. Again, this is because we made the management of forests by people who know how to manage it illegal. In the West, climate change is also contributing. Rising temperatures, earlier snowmelts, extended dry season, when the forests are then most vulnerable for the burning. Some ecosystems are adapted for less intense fires. Little is known about how plants or associated microbiomes respond to megafires, particularly in California's charismatic redwood tan oak forests. This is quoting Sydney Glassman, UCR mycologist, studies fungi, lead study author. It's not likely plants can recover from megafires without beneficial fungi that supply roots with nutrients or bacteria that transform extra carbon and nitrogen into post-fire soil. Understanding the microbes is key to any restoration effort. And it's sort of interesting because that kind of touches on what I was just talking about. You got the scientists who can understand the mechanisms in play. You have the indigenous population that may not have known about this mechanism, but knows what the result is if you do the control, right?
Yep.
Yeah. In addition to examining megafire effects on redwood forest microbes, the study is unusual for another reason. Soil samples were pulled from the same plots of land both before and immediately after the 2016 Sobranas fire in Monterey County. Get this kind of data, a researcher would almost have to burn the plot themselves. So it's tough to predict exactly where there will be a burn. Glassman said, sounds like maybe they had been just sampling this forest anyway, and then the fires happened, and they went, hey, let's go back and take a look.
Let's take a look. Yeah.
Yeah.
Opportunistic scientism.
70% decline in number of microbe species. And yet they were surprised that some yeast and bacteria not only survived the fire, but increased in abundance. So, which sort of makes sense. It's sort of like taking an antibiotic, if you think of it that way. It knocks
Right.
out almost everything. But if anything isn't getting knocked out, it now has no competition. It now has nothing preying on it or fighting with it or competing with it for those resources. And it can sort of blossom. Yeah. So massive increase in a fungal category for heat-resistant Basidioscus yeast, which is able to degrade different compounds of wood. This is part of
But
the part of the life cycle of the forest is to have these fires. And of course,
Yeah.
as we've also learned over these years, there are microbes and fungi in the soil that are beneficial to the growth of plants. One of those things, I think it's a long time ago, we talked about soil microbiomes in the frame of invasive plants, ones that start to take over, aren't just invading like the plant is very prolific and it keeps growing places, but it's actually the bacterial, the microbiome of that soil that is spreading, that is then allowing the plant to take over
Right.
from a native species.
Yeah. So after a fire, a big mega fire, you've got these species that they're like, that was great. And then they start making the soil better for the plants, to the first plants, the primary ones which come in, which are basically invasive. The
Mhm.
ones that come in first after a fire are like the quick growing, they're able to take over. Oh, that's
And this isn't in the study, but it's something that it was a research I'd read some time ago about native plants in California. They're more fire resistant than most plants. The
yeah.
the flowers like California wildflowers are more resistant to fire and heat than most. So California has been a fire state
For a
going back.
very
It's
long
not
time.
completely a new thing.
Yeah.
These mega fires aren't just a global warming thing. This is also just the way that California has always
Yeah. But it's good to understand this succession, how it works, and like you said, the microbiome aspect of it, how that actually works. This is This Week in Science. Thank you for tuning in tonight. We just have a few more stories to go. I have some brainy stories for us, as I love to do at the end of the show. Bring you stories that stimulate your brain about the brain. Um, New research just published out of the Tokyo University of Science. Researchers have been using a mouse model to study fear extinction. Now, this isn't like evolution and extinction. It is getting rid of fearful memories. And it would be a very important thing to be able to do. It's been under study to treat people with post traumatic stress disorder, to help remove fearful responses that occur in an out of place situation. These researchers in Tokyo have just published their work in the Frontiers in Behavioral Neuroscience, and they're looking at a compound called KNT127. It sounds just like a scary research drug that you would have like in, I don't know, some end of the world video game. But KNT127 has been shown to help mice get over fear conditioning. And so in the behavioral experiments they did with this compound, what they do is they get the mice into a, an area with a shock plate, and they
been.
are conditioned with a mild electrical shock to basically come to fear a certain place in an arena. And the conditioned fear response, unfortunately, is freezing. Mice don't fight or flight; they freeze when they have fear. And this question, though, they've shown that they are able to condition a fear response in mice and then give them the compound, and the compound causes the forgetting of that conditioned fear response. So the mice could then again go in the arena and not have any fear of potentially getting shocked. So it removes that association. How does it work though? Where does it work in the brain? The researchers had a drug that they had no clue how it worked, which is actually like a lot of things that we put into our bodies. Antidepressants, how do they work? No, we know a little bit about them, but the exact mechanisms are a little uncertain. So these researchers injected several brain regions: the basolateral nucleus of the amygdala, the hippocampus, the prelimbic and infralimbic regions of the medial prefrontal cortex, separately while they were re exposing mice to their fear conditioned stimulus. And what they found is there are two areas that they applied it to: the amygdala and the infralimbic subregions were specifically involved in extinguishing that fear response. And so, what they've discovered is that this compound selectively interacts with de-opioid antagonist receptors. So the opioid system, which is a natural system within the brain of receptors for pain and inflammation. And this KNT127 specifically, when it connects with these opioid receptors in the amygdala, which is, we know, an area of the brain responsible for our fear responses, that the KNT127 will get rid of the fear responses. So now we know where it's acting, which receptors it's acting on in the mouse brain. And hopefully they'll be taking this successful mouse drug and transferring it to human trials very soon.
So that's super important because like you're saying the amygdala is that primitive brain. I
Yeah.
like to refer to it as this primitive part of the brain that we don't have control of. It's part of the brain that thinks the brain is in control of us. Right? It's part of the brain that's
Mhm.
like, I need to tell this stupid bag of mostly water what to do, what to be afraid of, and how afraid to be. And if there's a problem there where it's not turning off, or it's too dialed up,
Yep.
there's nothing you can do about it but to go on drugs because you're going to have panic and fear and stress and all of these things. So being able to tackle that part of the brain, that's huge.
Yeah, yeah, it would be, and to be able to potentially use a compound like this, it's very specific to the area of the brain that it works in and can be used along with therapeutic means. So you've got a therapist and you have the application of the drug to help you have exposure therapy to get over your post traumatic stress disorder. It could really change people's lives moving forward. So it's also just interesting. Some areas of the little tiny areas of your midbrain are responsible for these fearful memories, and others are not quite as involved, which is pretty interesting, the specific areas. But how do they all connect together and what do they do? We've talked before about like these synchronization waves that happen within the brain and how you have like the firing and the wiring of neurons together in the brain to create memories, but it all happens within this greater context of synchronized oscillations within the brain, these greater brain waves. We've got our beta waves, delta waves, alpha waves, all these different kinds of waves that set the neurons to particular states to be firing or not firing, receptive or not receptive. A new study out this week, researchers have been trying to figure out how they could come up with computational methods to describe what's going on in the brain better than the methods that we've got right now. And right now, most of the neural networks that we have that we're making assumptions on, they are just like neurons turn on, neurons turn off. And they're like these single connections. And it's not as complex and it hasn't been able to incorporate these waves of brain activation that occur in different areas of the brain. Like, for example, in working memory, you have these circular oscillations of brain waves that kind of go woo, woo, woo. So they whirl
Oh,
around.
that's the sound I hear in my head all the
Yeah.
time.
They whirl around in your head. And researchers have been trying to figure out, wow, why is this circular oscillation happening in that area? Whereas other areas like the visual cortex have more of a parallel waves or more of like a single direction wave of activity through the brain. And then you have like the overall kind of like the tides, the overall brain waves that occur. So these researchers have given the analogy now that our brain is like an ocean with all of the waves that are interacting, and they're trying to come up with the complicated math to describe how it all works
Hmm.
so that they can create simulations that are more accurate, that may eventually be able to be applied to future artificial intelligence systems. And so yeah, this group out of the Salk Institute, they have been really this way of the nervous system, of the brain parsing information. It's not just a light switch on, light switch off. It's not just neuron on, neuron off. Your brain's going or not. That there are these interacting waves that they have interference patterns, and they can be additive or destructive. And so they're describing it very much more so like waves in an ocean.
Mm.
So your brain is like an ocean.
And that swirly bit you were talking about, it's like a gyre filled with
Gyre.
garbage in your brain.
No, it's like the phone number you're trying
Like
to remember.
catches the garbage, just keeps it. That's like when you're having anxiety, where you're just rethinking the same
Yeah.
garbage over. It's like the Pacific Garbage Patch.
The
It's all-
plastic,
Yes.
the plastic islands.
yeah.
But you can
The
imagine, like,
garbage
with
islands
working
in the middle of the
memory,
ocean.
that's like you're trying to keep something in your mind.
Mhm.
So it is like a gyre. It's like you're trying to keep that thought in your head. You're trying to remember something, like a phone number, an address, or, whatever it is you're working on. And so it's good that you have a gyre there to keep the thought there, keep the activation there, as opposed to, whoop, there it goes. Where'd it go? I don't know. The tide took it out. And then finally, adding on to this idea of our brain as an ocean that the Salk Institute researchers have been modeling, a group at MIT out of the Pickever Institute have been also looking at these wave interactions, but in the instance of the use of anesthesia. And so they just published a paper in the Journal of Cognitive Neuroscience in which they studied a compound called propofol, which is used very, very often for anesthesia, knocks people out, you lose consciousness. And the question is, how does it work? And we know that different anesthesias work differently, but this was a different way of looking at what was actually happening. And the researchers were able, through an 8x8 electrode array in which they were looking at an area of the brain, they were able to check out what these waves, a traveling wave moving through the visual system, and what ended up happening. And so with this little square 8x8 array of electrodes in this small little area of brain just trying to pick up a stimulus, they observed the normal activity of the brain, injected the propofol, and they then observed a wave of deactivation that pushed the activation that had been in an area out of the area. And so they saw, in a larger sense, that the propofol, like in an ocean, just kind of came in and got into the waves and took down the wave activity, interfered with the normal wave activity, and knocked it all down. So it was um, the propofol created a different situation to change the activation of the brain. And so, yeah, you might be going, yeah, the brain it's active or it's not active, right? The propofol knocks it out. Of course, it's just like an anesthetic. Of course, it's going to knock it out. But it's not that it knocked it out. It's the way in which it was knocked out. That it's not, again, like a light switch. That it is a traveling wave of deactivation through the brain. And so these researchers are saying that the rhythms we associate with higher cognition are drastically altered by propofol. The beta traveling waves seen during wakefulness are pushed aside, redirected by delta traveling waves that have been altered and made more powerful by the anesthetic. The deltas come through like a bull in a china shop. And delta activity is the kind of activity that you have in your brain more often when you're sleeping. Beta activity is your wakeful, fast moving waves, and the delta activity is the big, slow moving stuff. And so the Delta bull in a china shop of propofol comes through and says, "Oh, it's my turn." So the question now is, they want to look at other anesthetics that are being used to try and determine whether or not these slow frequency delta waves are traveling in the same way when you use different types of anesthesia. Is it always the same? Is it different? How do these waves interfere or add to influence consciousness itself? We have made it to the end of another show. It took us a little while to get here, but we are here, and we are so glad that you stayed with us until the very end. Thank you for joining us for another episode of This Week in Science. I do want to thank a few people who are important for making this show happen. Time for shout-outs. Shout-outs to Fada. Thank you so much for your help with show notes and social media. Couldn't do this without you. Identity4, thank you for recording the show. Gord and Arnlore and others who help keep the chat room happy and kind. Thank you for being here and doing that. And Rachel, thank you for your editing and other assistance. And many thanks to our Patreon sponsors. Thank you to Teresa Smith, James Schofer, Richard Badge, Kent Northcote, Rick Loveman, Pierre Velazar, Ralphie Figueroa, John Ratnaswamy, Carl Kornfeld, Karen Tozzi, Woody MS, Chris Wozniak, Dave Bunn, Vegard Shefstad, Hal Snyder, Donathan Stiles, a.k.a. Don Stylo, John Lee, Ali Coffin, Maddie Perrin, Gaurav Sharma, Ragan, Don Mundis, Stephen Alberon, Daryl Myshak, Stu Pollock, Andrew Swanson, Fredis 104, Sky Luke, Paul Ronovich, Kevin Reardon, Noodles Jack, Brian Carrington, Matt Bass, Sean and Nina Lamb, John McKee, Greg Riley, Mark Hessenflow, Gene Tellier, Steve Leisman, a.k.a. Zima, Ken Hayes, Howard Tan, Christopher Rabin, Dana Pearson, Richard, Brendan Minish, Johnny Gridley, Rami Day, Flying Out, Christopher Dreyer, RDM, Greg Briggs, John Atwood, Rudy Garcia, Dave Wilkinson, Rodney Lewis, Paul, Philip, Shane, Kurt, Larson, Craig, Landon, Mountain Sloth, Jim DePaul, I can't go any higher, Sue Doster, Jason Olds, Dave Neighbor, Eric Nappy, Kevin Parachan, Aaron Luthi, Steve DeBell, Bob Calder, Marjorie Paul, Disney, David Simmerly, Patrick Pecoraro, Tony Steele, and Jason Roberts. Thank you for all of your support of TWIS on Patreon. And if you are interested in supporting us on Patreon, head over to twist.org and click that Patreon link. On next week's show, we have an extra special interview next week, which I hope that you'll be able to join us for next Friday, May 6th at 11 a.m. Pacific time with Dr. Gregory Forth on the Hobbits of Flores.
Homo floriensis.
Nice. Yeah. But what do we do most Wednesdays, Justin?
Well, we'll also be back for the regular show Wednesday, 8 p.m. Pacific time, broadcasting live from our YouTube and Facebook channels and from twist.org/live.
Hey, do you want to listen to us as a podcast? And maybe while you make some chocolate sourdough bread, since you got sick of all the normal sourdough you made during lockdown, just search for This Week in Science wherever podcasts are found. If you enjoyed the show, get your friends to subscribe as well.
For more information on anything you've heard here today, show notes, links to stories are available on our website, www.twist.org.
You can contact us directly. Email Kirsten at kirsten@thisweekinscience.com, Justin at twistminion@gmail.com, or me, Blair, at blairbaz@twist.org. Just be sure to put TWIS, TWIS, in the subject line, or your email will be catapulted away from some orb weaver spiders, which is good, but it will also be away from us, and we will never read them.
You can also hit us up on the Tesla, where we are at Twist Science, at Dr. Kiki, at Jackson Fly, and at Blair's Menagerie. We love your feedback. If there's a topic you'd like us to cover or address, a suggestion for an interview, a haiku that comes to you in the night, please let us know.
We'll be back here next week, and we hope you'll join us again for more great science news.
And if you've learned anything from the show, remember,
it's all in your head.
This Week in Science. This Week in Science. This Week in Science.
This week in science, it's the end of the world. So I'm setting up shop, got my banner unfurled. It says the scientist is in, I'm gonna sell my advice. Show them how to stop the robots with a simple device. I'll reverse global warming with a wave of my hand. And all it'll cost you is a couple of grand. 'Cause this week's science is coming your way. So everybody listen to what I say. I use the scientific method for all that it's worth. And I'll broadcast my opinion all over the earth. 'Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in 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 Science.